Good morning, students. I hope you are doing well. So, students, today we will discuss the topic therapeutic drug monitoring. In that, first of all, we should know what is therapeutic drug monitoring. It is nothing but the monitoring when we are doing any course of therapy, when you are doing any clinical trial. At that time, we are checking that blood level of that drug into the patient's body, so that we can evaluate that whatever drug we are giving, it has been reached into the circulation or not. So for that we are doing this therapeutic drug monitoring. So what drugs can be monitored when the range between minimum effectiveness and toxic is narrow? Means there are certain drugs which are having very less range between effectiveness and toxic effects. Means suppose their effective dose is minimum effective dose is suppose 100 mg, and that drug's toxic dose will be 120 mg, means range is only 20 mg. So due to that narrow therapeutic range, we have to give this drug very very consciously, so that it should not reach to the highest toxic dose. Means it should be between 100 and 120. So that should maintain into that dose, so that it should not be decreased that it won't get any, it won't show any effects, and it should not be increased that it will start to show its toxic effects. There are certain drugs that I would like to discuss with you. The drugs like phenotine, its range is 50 to 100 mg per liter. Next drug is digitoxin. Its dose is zero point 0.8 to 2 microgram per liter. So at which dose it should be maintained. So like this, there are n number of drugs in which we have to give them under control. The result of the drugs assay provides significant information of the clinical decision making. According to their assay, we can evaluate that what is their effects, at which dose we can give them, and by that dose it will start to show its toxic effects. Now, why TDM is required? Why there is a role of TDM? Now, first role is non-compliance. What is non-compliance? When any patient is not able to go with your medicines, if patient don't like medicines or he start to avoid medicines, means non-adherence with the drug. It is nothing but the non-compliance. So there is role of pharmacist to convince the patients to take that drug. So here the non-compliance is the major issue to in our TDM. So that's why it is our role to check. Suppose the patient is telling that that he have taken that drug. So by taking its blood sample, we can check the level of drug, the concentration of drug in his blood, and by this way we can understand the patient is telling lie or not. So here non-compliance is the major issue for your loss of TDM or drug loss, because if no drug is there in his blood, means patient is not taking drug, he is avoiding or he is keeping the doses. Next problem is inappropriate doses. Sometimes patient won't get the appropriate dose of that typical drug. It occurs especially in case of the liquid preparations, when there is not single dose drug is available. In case of tablet or capsule, it is it is possible that we are taking proper dose, but in case of liquid dosage forms, or in which the typical proportion is in our hand, that it completely depends on the patients. So when patient is doing mistake in taking that dose, So it will also leads into the toxicity. Next is poor availability. This is also one of the problem, because not all the bodies are showing the same response. That depends on the person to person variation. So some people are showing good bioavailability, and in some patients you will get less bioavailability. So that will also checked by doing this TDM. Suppose TDM, if you are started, so we have to take the blood sample according to our protocols, according to our SOP. Either we are taking the samples in 24 hours, or we are taking in 12 hours or 6 hours, and in some critical conditions we are taking in 3 hours also. That depends on a protocol and on which disease you are querying. Accordingly we had to decide our TDM. Another issue is drug interactions. When one drug is going to alter the effect of another drug, it is called as drug interactions. And due to that also, there will be less bioavailability, less concentration of drug will be available into our blood. That's why you should ensure that there is no any drug interaction, so that it will reduce its concentration of drug into the blood. Next is kidney and liver disease. These will also affects on our drugs effects and its concentrations. Suppose your kidney is working too much fastly. If any kidney failure occurs, renal failure will occurs. So due to that also, what will happen? The drug won't stay into your body for longer period. It will be immediately washed out. It will throw out from the kidney, because what is kidney? Kidney is the major source of drug excretion. Whatever drug is going to be excrete, out of them 80 to 90% of your drug is going to be excrete through our kidney. That is one issue. If drug will go away from our body very fastly, it will also decline its concentration. And another is liver disease. What is the role of liver? Liver is going to be biotransform our drug. It is responsible for our metabolism. If the metabolism of rate of the liver is going to be declined due to some liver disease like fatty acids or liver fibrosis or any sort of liver disease, so what will happen if liver is having any disease? Liver will do malfunction. Liver will work slowly, or there is disturbance into the metabolism process, biotransformation process, and due to which the drug which is available into our body, in our blood, it will metabolize slowly, and due to that it will stay in your body for longer period, and its blood concentration also will be increased, and due to that high concentration the drug will start to show its toxic effects, and which is not expected. That's why in case of liver disease also you should give the drug very very consciously. You should, like a watchdog, you have to see, you have to watch each and every movement, that suddenly if the blood concentration of that drug will be start to increase or start to sustain, in that we have to stop the dose, or we have to release the drug, or we have to decrease their concentration through blood. So here again the monitoring is very very important. So dear students, here we are discussing this TDM, therapeutic drug monitoring, very shortly, because already we have covered this TDM in last lectures. We have taken this topic very in descriptive manner. So that's why in today's lecture we will discuss it in a short manner, because it is only a repetition. So next point is altered protein binding. Here, what is the issue? There are certain drugs which will alter the protein binding of that drug. For example, warfarin. Warfarin sodium is the drug, and also there are n number of drugs. They are affecting on the protein binding of your drug. Suppose if your protein binding is more, what will happen? The drug which has been reached to your plasma concentration will be increased. So that's why the drugs which are altering, if you are giving any drug along with the warfarin, means they are they are dispatching or they are separating or removing the drug from the protein binding, and they will do the displacement of that drug. Next is fever. In fever condition also, it will affect the blood concentration. And cytokines. What are cytokines? They are nothing but the peptides, proteins. And these proteins are in the form of interleukin or interferons. They are the cytokines. And when they are released at a time of inflammation. They are released at time of immune response. And who release these cytokines? The cytokines are released by the macrophages or T cells or lymphocytes. They are responsible for release of these cytokines. Cytokines are released in the form of signaling. Whenever any sort of pathogen will attack in your body, if they will attach to your cell, at the time the cytokines will be released by these lymphocytes or macrophages in the form of signaling. So they are nothing but the interferons, interleukins 1, 2, 3, 4, 5, 6, interferons 1, 2, 3, like that they will release. And whenever the next dispose or the next exposure of the same pathogen will be there, at that time again these cytokines will be released, means they are responsible for the immune response or developing of the immunity. They are signaling and they are identifying that typical sort of bacteria or viruses. So cytokines. And when the cytokines are released, so they will also affect on your blood concentration of that typical drug. Next is genetically determined fast or slow metabolisms. What is this? These are nothing but the microsomal isoenzymes which are available into our liver, and they are genetically determined. They are specific list of microsomal isoenzymes which are available into our body according to our genes genetically. That's why according to our continents, according to our race genes, the typical microsomal isoenzymes are available into our blood. That's why they are genetically develop the metabolizing ability for typical drugs, because due to that they are deciding we are having slow metabolism or fast metabolism. Suppose if one microsomal isoenzymes is available in some genetically people, due to which a drug will be metabolized, and suppose the the B drug, for B drug whatever the isoenzymes is responsible for its metabolism, if it is not available in that typical genetically peoples, due to which that drug is not going to be metabolized. Understood or not? For metabolism of a drug, if that microsomal isoenzyme is available, means that A drug will be metabolized. And for B drug, if that isoenzyme is not available, means that B drug won't going to be metabolized. And due to which, what will happen? That B drug will remain in his body for longer period, and it will start to show its toxic effects, and it is very dangerous things. So that's why mostly the drugs which are having narrow therapeutic range, they should be taken very very consciously. And also according to the genes, which drugs are more suitable for that typical genetic peoples, that we can identify. Now, students, from these slides, we will start to discuss the responsibility of clinical pharmacist. What are the responsibilities? Now, in that first is designing patient-specific drug dosage regimen. As recently we have discussed, according to the genes, each person is having their own microsomal isoenzymes in their body, and each person is having their specific sensitivity for specific drug. That's why for specific person we have to define the specific drug and specific dose regimen. drug as well as dose for typical person, so that we will get the proper therapeutic effect or therapeutic dose. Next is recommending or scheduling measurement of the drug concentration in biological fluids, that constantly we are doing. That is a major responsibility of that pharmacist. He must recommend to the patient to check his concentration of the drug into the blood, or you can check that concentration into the swab, or you can check in saliva, or you can check in mucus, any biological fluids. Mostly we are checking in bloods its concentration, means the drug is reaching where, whether drug is reaching your mucus, reaching your saliva, reaching your blood, reaching your urine, reaching your brain, at which body fluid it is reaching, it's reaching your lymph, areas of body fluid. So we can check, so we have to recommend to the patients to check its concentrations. Next point is monitoring and adjusting dosage regimen. That is also very important. According to the concentration of blood reaches to our blood circulation, concentration of drug into our blood. If more drug is available into your blood already, so accordingly we can modify the dosage and your dose. So whatever regimen we are giving, so according to their bioavailability, we can define, we can adjust the dose, and also the health status of the patients. If patient's health status is weak, so accordingly also we can adjust the dose, we can decrease the dose. Or if health status, if the person is deceased person, in that condition also we can adjust the dose. Constantly we should monitor the dose. Evaluating unusual patient response to drug therapy, that is very important. If any person is having any adverse events or adverse drug reactions or adverse effects, in that case we should monitor them, and for possible pharmacokinetic and pharmacological explanations. So once you got any sort of adverse drug reactions for any pharmacokinetic effects, so we should able to explain the proper reasoning. So what was the reason? Suppose if any person is having fever or skin rash or any sort of complications, so we must have the proper reasoning. What will be the pharmacological reason for that? For which reason that effects we got? So immediately we should come to conclusion that we should identify, we should find out the cause of that typical adverse effects, and also we should check in how many patients the adverse effects we have got. If we got only one patient, so we should monitor that patient, we should identify, try to recall and check out, find out the mistakes where we caught. So in this fashion we should monitor that. Now next responsibility, communicating patient specific drug therapy information to physicians, nurses, and other clinical practitioners, and to patients orally and in writing, and including documentation on this in the patient's health record. Here, communicating the patient specific drug therapy. So we should communicate that this is a typical patient, this is having typical disease, or this is the typical health status, or the patient is having any sort of allergies. So we should categorize that patient, separate that patient, and we should discuss his condition with the physician or nurses or other clinical staff who are in touch with the patients, and also keep it in writing also. In writing, in database we should write, and you should highlight that this patient is having this problem, and we should uh, keep under the consideration that while giving any therapy, these are the parameters that we should consider and we should think when you are giving any therapy. And also you should maintain the record, constant record of certain type of patients. Next responsibility is educating pharmacists, physician, nurses, and other clinical practitioners about pharmacokinetic principles So here the role of that clinical pharmacist, that he must always educate the physicians or nurses or other staffs who are helping them, or also pathological staffs. So he must educate, he must tell them regarding the protocol, regarding the information, regarding the therapy, if any uh, parameter, if he are decided that for these patients this parameter we will follow. So he can educate, he can guide to the nursing staff, to the physician, to the consultant regarding their treatments. An appropriate indication for clinical pharmacokinetic monitoring, and he should give the appropriate indication, if any suggestion, if any sort of advice or any important points he want to tell while doing the pharmacokinetic monitoring. So he should also put that information into these peoples, including cost effective use of drug concentration measurement, and also including the drugs which are cost effective, which are having less price than the normal. So we are also thinking in that way, and we will give more concentration of these pharmacoeconomics drugs, by which we can develop certain drugs which are having less cost for the peoples who cannot buy them at high cost. Next is developing quality assurance programs for documenting approved patients' outcomes and economic benefits. It is necessary that we should develop the QA programs, because whatever the clinical trial we are doing, whether it is producing any outcome or not, whether it is cost effective or not, that we should find out its quality, we should find out its outcomes. Whatever money we are pouring them, whatever money we are investing into this clinical trial, because we are investing crores of money into one clinical trial, and suppose if you are getting nothing, means we are wasting our money. So it should not happen. So periodically quality checking, quality assurance we should do during the clinical trial. We should check during clinical trial from beginning and to the end. Constantly we should monitor its assurance, its quality, whether it is going in good direction or not, whether all the people are doing their work properly or not. That we should ensure. Promoting collaborative relationship with other individuals and departments involved into the drug therapy. That is very important. If all the departments are work collaboratively with cooperation, means there is 100% chances of success. Otherwise, what will happen if one department is having issue with another department? They won't cooperate. They won't discuss their facts, and they won't work collaboratively, combinedly, and we won't get the expected results. So for that, all the departments should work like a team, like a good team, so that we should able to get good results, because we are investing money, too much money in that, and for that we cannot compromise on any sort of mistakes or any errors that will happen due to the issue into our staffs. So that matter should be solved. Another responsibility: pharmacists with specialized education, training and experience may have opportunity to assume following additional responsibilities. The first is designing and conducting research. Yes, because clinical pharmacists are born to do the research, because they are having that capabilities. So they can design any sort of protocol, and they can conduct the research. Second is developing and applying computer programs and point of care information system to enhance the accuracy and sophistication of pharmacokinetic modeling and application to the pharmaceutical care. Yes, because computers are very accurate into their work. So why should not we should take use of these computers? So we should use it for increase our accuracy and to do our work more sophisticatedly. because in pharmacokinetic modeling, if you will use computers, so we will get more specific results, and we can compare our results with previous results, and by that way we can enhance our work. Next responsibility is serving as an expert consultant to pharmacist with general background in clinical pharmacokinetic monitoring. Yes, we can become expert to the pharmacist, to the community pharmacist who are working into the community, who are in touch with the general peoples. So we can give them experience, we can share our experience, we can give them more knowledge regarding our clinical trial experience, we can educate them, we can make them expert into their work. So like that, these informations of tdm or clinical trial, who are doing this, done by this principal investigator who is a clinical pharmacist, so they can concerned with the pharmacist, they can teach them. And in this fashion we have discussed this point today. So students, if you have any queries, you can ask me. Like these responsibilities are there of the pharmacist, so today we have discussed. Now there are again few points that we'll discuss only in 1 or 2 minutes. This point, medical record. So from where we are getting the medical record means from where we are getting the medical information. So in that admission information, once the patient has been entered into the enrollment of our program into a clinical trial, he's giving the information like age, sex, patient's name, his history, all these things, and initial history. If patient is having any symptoms, initial history we are getting. Physical examination by checking the patient body temperature, his uh, his eyes or his hand, his chest by stethoscope. Any physical examination, we are checking temperature like that, or blood pressure. So by which we are getting the medical information. Progress notes means whatever progress notes the nursing staff is constantly visiting, or pharmacist is constantly visiting to the patients, and they are writing the progress notes regarding the blood pressure or their pulse rate or oxygen consumptions. So like that information they are writing, or their body weight, if they increase decrease in body weight. So that progress note also is one of the source of medical record. Consultations, we doing discussion with the patients regarding the DCS. It is also consultations. We are asking the questionnaires to the patients. And nursing notes, nurses are visiting to the patients and they are getting the informations. And laboratory data, the pathological data which we are getting from the pathological lab. So by that also we are getting the information regarding the disease, what sort of disease or what sort of problem the patient is having regarding RBC, WBC, or any sort of pathologic or any bacteria virus is available, or any sort of incline decline in his normal values, that we can identify. Diagnostic procedures, any sort of diagnostic tools we are using. For example, in case of infertility, we can check the uterus of the woman by some testing like HSG. Like next is radiology. By doing the radiology of X-ray of chest, we can identify how many mucus has been developed or the pus is there or not. That we can check. And surgery, by doing surgery also we can get the information, orders, medication administration orders, and consent form. What is consent form? The consent form which has been filled by the healthy volunteers at a time of enrollment. The consent form means that is the letter or consent or his willingness, patient's willingness to enroll into the clinical trial by his own. There is no pressure of any authority to enroll into the clinical trial. That is nothing but the consent form. So like this, all the informations for a medical record we're getting. So here we will stop and we will continue the session in next lecture. Today we have completed this point. If you have any queries, you can ask me. So thanks a lot for hearing me and have a nice day. Bye.